on derivatives of monosaccharides. So, monosaccharides can react with many other molecules, many other substances because it has OH group present where the oxygen is electronegative and forms the derivatives of the monosaccharides. Okay. So, the derivatives of the monosaccharides are phosphoric acid ester of monosaccharides, amino sugars, deoxy sugars, sugar acid, sugar alcohol, uramic acid and sialic acid. So let us now move on to the derivatives of monosaccharides in detail. First of all, we will be starting with the phosphoric acid esters of monosaccharide. We already know that there is the OH group present at the first position of the glucose. Okay, so whenever the OH group of the sugar, that is the hydroxyl group of the sugar, it reacts with the phosphoric acid, then the phosphoric acid esters are formed. All right. So why these phosphoric acid esters are formed? Here you can see this is glucose 1 phosphate. The phosphate group is being attached on glucose, the first position of glucose. Here is the glucose 6 phosphate. The phosphate group is attached to the sixth carbon atom of the glucose. All right. So now let us understand that why this bonding or this phosphorylation of sugar is important. Why it is important? First of all, when the glucose it enters into the cell and it prepares itself for glycolysis, as soon as it will enter into the cell, the phosphorylation happens. It gets converted into glucose 6 phosphate. So now why this phosphorylation is important? It is important because so that the glucose it should not go along with the concentration gradient outside the cell. It should retain inside the cell. The glucose molecule should retain inside the cell. So when it must retain inside the cell, so then the further it will be utilized for energy in glycolysis. And secondly, the nucleic acids such as DNA, RNA, we already know that the DNA, RNA is composed of phosphate and sugar backbone. And what are the sugars present in the DNA and RNA? They are the ribose and the deoxyribose. Okay, so let us move on to the amino sugars. In amino sugars, the hydroxyl group, it is replaced by either amino or acetyl amino group. The examples are glucosamine, N-acetyl glucosamine, galactosamine and mannose-amine. So this is the structure of N-acetyl glucosamine. Here you can see that the hydroxyl group here is replaced by an acetyl group. Okay, so now why these amino sugars they are important? They are important because they are important components of glycolipids, they are important components of glycoproteins, they are important components of antibiotics such as erythromycin, such as carbomycin, and hence they are very important. Now moving on to the deoxy sugars. So the deoxy sugars, they possess a hydrogen atom in place of one of their hydroxyl group. So if we generally see the structure of ribose sugar, here in the ribose sugar, here at the second position there is HOH, but here you can see the oxygen is removed, hence it is a deoxy sugar, and the oxygen is removed at the second carbon atom, from the second carbon atom, hence it is 2' deoxy sugar. When it's the 2' deoxy sugar, and it's important component of the DNA. Moreover, there are some sugar acids that are produced by the oxidation of monosaccharides. For example, glucuronic acid. Here the CH2 OH group, it has been replaced by the carboxylic acid group, and hence it becomes the glucuronic acid. Further, there are also derivatives which are alcohols of the sugars. Okay, so they are not metabolically very much active, but they have certain medical importance, and they can be used in certain foodstuffs for diabetics, such as sorbitol and xylitol. Okay, so next, moving on to the neuramic acid. This neuramic acid, it is the 9 carbon sugar which is derived from the mannose and pyruvate. So mannose and pyruvate, it forms the neuramic acid. Okay, so that's all for today. Please correct the spelling. This is neuramic acid. Neuramic acid. Okay, all for today. And in case if you want, lecture on any other topic, please comment down in link. Thank you so much. Stay tuned.